right, everyone. Welcome to the final unit of sugars and nucleic acids. Today we're going to talk about nucleic acids and their applications for things like therapeutics, how we can use chemical modifications of these oligonucleotides to improve their properties as well. So it's divided up into 5 main sections. I'll talk very briefly about chemical modifications broadly and where we can put them within oligonucleotides, and then we'll transition to different applications like antisense technology, siRNA technology, aptamer design, and then how we can use some biophysical tools to study oligonucleotides. So you just finished the unit on solid phase DNA and RNA synthesis, and you learned that this is a cyclic cycle in which you detritiolate and then you add your phosphoramidite as shown here. You couple this with typically an activating agent like tetrazole. You generate a phosphite linkage which you can then oxidize and continue the cycle, so on and so forth. So let's take a closer look at what a DNA or RNA synthesizer looks like. This is a picture of a machine called a DNA synthesizer that's actually very similar to the one I have in lab. So what you can see here is that you have a number of different vials on top, and then you have a number of different solutions on the bottom. It's a little hard to read what these say, but this here is the activator solution. So this would be something like tetrazole or S-thioltetrazole. This bigger bottle here is actually 3% trichloroacetic acid. So that's involved in the detritiolation. And these other solutions here correspond to things that you've seen before, like the iodine, oxidation, capping solution, so on and so forth. What you have up here are the phosphoramidites. And you can see here we have a spot for A, a spot for G, spot for C and T. And these machines that were built, this is actually an applied biosystems synthesizer. So this is an older one. This one has room for, other phosphoramidites. Okay, so maybe they are RNAs, or maybe they are different modifications that a researcher might make in lab and then want to use these building blocks to synthesize a desired strand. What you have here is a solid support. The solid support is where you do and conduct the synthesis, and then you have these lines here that will push the solvent through in reagents, and then essentially it's a cartridge which will then allow excess waste and solvent to collect. So at the end of the day, you would have a fairly pure product in the cartridge, and then you would deprotect that with ammonium hydroxide, as you've learned. You have a number of keypad buttons here, and you literally just program your sequence in. You can modify the program in case you want to increase things like coupling times, or if you want to do some other types of engineering or modification. You can't see this, but this corresponds to AGCT. So if you punch these into your sequence that you want to make, then these would correspond to the corresponding phosphoramidites. These phosphoramidites you can also purchase from chemical suppliers like CHEM genes. So I would encourage you to Google something like CHEM genes. There's other companies as well throughout that sell these building blocks. So we don't necessarily always have to make them. We can actually purchase them. If there's something novel that we're making and no one's made before, then obviously that's not going to be commercially available. What else? Here we have gas. What does gas mean? So we keep this system, this DNA synthesizer, under pressurized inert gas. So typically something like argon or nitrogen gas. And the reason we do this is that it's really important that we avoid moisture in the system. So we have to use anhydrous solvents. We want to exclude moisture as much as possible. We typically keep this at around 50 to 60 psi's. And the reason we want to exclude moisture is because if we go back to the synthesizer step, during that coupling step, where you get really good yields, you have a primary hydroxyl group reacting with the phosphoramidite. However, if you do have water in the system, that will compete with this reaction, lower your coupling yields, and therefore lowering your overall yield, especially as you start making longer and longer oligonucleotides. So it's always good to do DNA synthesis in areas where it's not humid. Sometimes in the summertime, if we are making oligonucleotides and it's a very humid day, perhaps a building has trouble with the air conditioning, we actually don't use it because we want to avoid any type of moisture getting into the system. All right, so that's a little bit of background on the machine and how it works. Let's talk a little bit about sites of chemical modification on oligonucleotides. So you've learned that oligos are made of base, sugar, and a phosphate. And areas that researchers typically like to modify include areas such as the sugar. Oftentimes people will modify the base, and oftentimes people will modify the backbone. So sometimes people will research and modify one particular area, or they'll do this in combination. And the reasons researchers want to do this is to increase the properties of these oligonucleotides. They want to give them some inherent strength, perhaps increasing their capacity to cross cellular membranes, so perhaps to increase its delivery methods, perhaps to increase its hydrogen bond capabilities to its partner, to increase its bonding affinity. So there's a lot of things that researchers can do with modifications, and I'm not going to go through any specific ones per se, but I just want you to know that analogous to how we looked at peptides, peptoids, and some of those modifications, you can apply those same strategies like you would for oligonucleotides. These are just some examples of some modifications, some sugar modifications that have been studied. And I'm just going to highlight typical RNA looks like this, DNA looks like this with a deoxy sugar. But researchers have looked at things like O-methyl groups, fluorinated groups, the impact that a fluorine atom has. We've looked at fluorine atoms with respect to peptides also and peptoids. Fluorine in a different position, how that would impact the sugar. There, you can see here that they come in many flavors. Now we have a bridging sulfur group. What effect would that have? We have things like this unbranched nucleic acids where you don't have a bridging carbon between the C2' and the C3' position. Other interesting ones are things like locked nucleic acids, where you now have a bridging carbon between this oxygen and this carbon. And we'll talk more about that if you take chemistry 4120 with me next term. So I won't go through all these different ones, but you can see that you can let your imagination run wild, and you can literally think of different modifications that would improve the properties of these oligos. And researchers have placed a lot of effort at modifying 2' positions with a variety of different groups. You can also change the size of the sugar. Here you have a 7-membered ring. Here you have the 6-membered ring that doesn't really look anything like a ribose or a deoxyribose. So it's really interesting to see what effects these have, not only biophysically, but how they could be used for various applications as well. Since we're going to be focusing a lot on applications, in particular therapeutic applications, it's important for us to understand how we can use chemistry within oligonucleotides to regulate gene expression. So if we look back to the central dogma of biology, we understand that the first committed step involves the transcription of DNA to messenger RNA, and then the messenger RNA is exported to the cytoplasm, and then the ribosome translates that particular messenger RNA to generate particular proteins. And you learn how this happens biologically too, and you understand the mechanism of how all this happens. So we have roughly 30,000 different genes, which means we have roughly 30,000 different proteins. And these genes are turned on or off at very specific times. So it's important to realize that they're not always on, and there's a controlled mechanism in place in the cell that clearly articulates which genes are turned on at which points. And different cell types would have different types of gene expression. Unfortunately, in diseases like cancer or other diseases like genetic diseases or metabolic diseases, you can have what is known as unregulated or aberrant gene expression. What this means is that the cell has lost the ability to control how much transcription and translation is happening. And what can happen is that you can get either an underabundance or an overabundance of proteins that are characteristic of that particular disease. Now, a lot of the drugs on the market, most of them are small molecules, and most of them target proteins, right? So we talked a little bit about this, if you recall, way back to our first introductory lecture, in which we looked at things like aspirin or naproxen, Advil, ibuprofen, and understood how these can actually inhibit a COX-2 and COX-1 proteins to avoid things like inflammation. So that was one example. However, there are many proteins out there that are responsible for diseases that are what we call non-druggable. So it's been estimated that around only 15% of proteins are druggable with small molecules. And even that is a fairly ambitious number. So what this means is that the 30,000 different proteins that we could potentially target with small molecules, that really comes down to a much smaller number. And that leaves lots of proteins that we could potentially target that we can't use small molecules for. So oligonucleotides actually have the potential to target not the protein itself, but different areas before the protein's made. So one area that we'll look at is called antisense mediated gene repression. And what it does is it targets the messenger RNA before it's translated. So you can think of this as a preventative type of way to remove the protein from the cell. Another way that we'll talk about next also is short interfering RNA, and this also targets the messenger RNA before it is made. We'll also look at aptamers. Aptamers are specific nucleic acid polymers that have high affinity for particular structures. So this could be something like a small molecule, or it could even be something like a protein. And there are a couple aptamer nucleic acids that target specific proteins. And then furthermore, there could be different ways to target the translational machinery, so trying to block translation from happening at the ribosomal site. We could even add a fifth one, in which is it possible to block transcription with nucleic acid analogs or things that look like them that would therefore perhaps block DNA transcription or mRNA transcription. So that just gives you a sense of what oligonucleotides can do. And what's really interesting is that the chemistry actually was first in line in terms of what it could be doing and how it could be synthesized. So this whole concept of nucleic acids as drugs, as I just showed on the last slide, received a lot of attention back in the 1970s. This generated a lot of excitement in the therapeutic field. And then shortly after, in the 1980s, efficient methods for DNA and RNA synthesis were developed. The first companies were founded, and the chemistry, as you learned, is really quite robust. So that was actually a fairly long time ago. Now, fast forward to around the late 90s, there was a little bit of good news, but not much. So a lot of the companies were founded, but only one approved drug was actually made it in the 90s. The bad news on many clinical oligonucleotides failed. So lots of money, a lot of confidence was lost in the industry to develop oligonucleotides as therapeutics. In the early 2000s, RNA interference, which is what we can use siRNAs for, which I'll talk about in part C, enters a picture. And there was really a lot of excitement because this uses an endogenous biological pathway to potentially target any messenger RNA. And there was a lot of progress with using chemical modifications to improve the properties of these siRNAs. However, after about a decade of work, there was again a steady stream of clinical failures. So 2010 came around. Now, this is when we learned a lot about the clinical failures, not only from antisense, but also siRNAs, and new approvals with nucleic acid drugs started to happen. This also included work with antisense oligonucleotides and RNAi drugs. So this was actually a very exciting last decade. However, still there's a lot more work that needs to be done. Currently, we are in the 2020s, and one of the big companies, Alnylam, they have a large platform of siRNAs in clinical trials. 2 of them are approved, but in general, what's nice is that there's healthy optimism in the field in which we can use these drugs to target various different types of diseases, including perhaps even using an siRNA to target the COVID-19 SARS-CoV-2 virus. Okay, so that's all I wanted to talk about in this short introductory lecture. The next unit will talk about antisense. See you then.